the slides?
Yes, it does.
All right. Thank you for the invitation to speak. I'm really excited today to share a new direction in the lab that I started with new collaborators at Melbourne University after joining. So I'd like to introduce, we don't have a lot of data on this yet, but we're super excited about the future. And I'd like to introduce the concept of why we set up a visual vertebrate pipeline. And so the idea, this is the brainchild of Pat Goodborn, Dr. Goodborn from the Melbourne School of Psychology. And he's really interested in using, he's a human visual neuroscientist, and he's interested in using the visual system to model different neurological diseases. And so my research is focused on the visual system, but we also do this with the view in mind that the visual system, which can be quite easily tested non-invasively in humans, might act as a good testing ground, because a lot of complex neurological disorders have a visual endophenotype. So to show that as an example, I've got here a visual stimulus. This is called a random dot motion stimulus. It's obviously visual, and hopefully most of you will be able to compute the motion of the individual dots to see a rotating drum. And it will change direction as well. Now, to show you how that visual stimulus can be related with other neurological disorders, once we introduce noise into the system, so make some of the dots behave more randomly, we find that, for example, schizophrenic patients have a harder time to see the global motion because they can't do that integration. So that's just one example of how we think about the visual system as a readout for central nervous system function. The model we're using for this pipeline, a key requirement was that it would be high throughput. And so my lab works on zebrafish, and Pat approached me to see if we could set something up in zebrafish. The key advantage for the high throughput part is obviously that the zebrafish can lay lots and lots of eggs. So here is a female zebrafish over the hundreds of eggs she's just laid. And because they develop outside of the mother, this allows us to quite easily perform molecular manipulation, so gene functional study by microinjections, and we can also very easily generate, of course, CRISPR mutants to model specific human genes. The other key advantage is that they develop really rapidly. And so you can see in this little time lapse that shows the first 19 hours of development, how rapid the changes occurs from the movie started at the 2-cell stage. You can see lots of cell divisions followed by gastrulation events. And before the first day is over, we can already see the basic vertebrate body plan has formed. So you can see the vertebrate column, the tail, and of interest to me, particularly the eye primordium has already started working. So all of the phenotypic analysis I'm showing today, except for the later part where I will talk a little bit about older fish, is done at 5 days post-fertilization. Now, studying the fish is not going to be very interesting if we don't have conservation across to what we're really interested in, which is understanding human health. So this Venn diagram just shows genetic conservation across different vertebrate model systems. And just to highlight that over 10,500 genes are highly conserved across the vertebrates. But also what I find interesting is that the number of genes that make each of these species unique is actually a similar order of magnitude. So the number of genes that make humans humans and different from mice is not that different from the number of genes that make zebrafish quite specific. Most of the work I'm showing was done by Jiaheng Shi, who did a master's with us and is now currently a PhD student. And again, the idea was to address a bottleneck that we're facing with a lot of the studies, because now that we can quite easily get lots of data from human genome-wide association studies, the question is then, what do we do with the hundreds of novel genes identified that are associated with a particular disease? And so that's where the high throughput screen comes in, where we want to use a high throughput system to model variants of unknown significance to identify perhaps the 10, 15 top genes that have an effect size that we can then take into the mammalian model. So this is sort of the first step or complementary to the disease modeling we would then expect to be done in animals that are more conserved with humans. So we're using the zebrafish, and the first step was that we really wanted this to go cross-disciplinary. So rather than just focusing on anatomy, we wanted to integrate electrophysiology and behavioral tests. And we wanted to correlate how different phenotypes match across these systems. And we think that's going to be really nice across different disorders, for example, to have a baseline with always the same tests done that can be compared. So the first task for Jiaheng was to set up the electrophysiology and behavioral tests on larval zebrafish. I'll just take you through the few slides through the analysis pipeline readouts. So my lab historically has primarily done neuroanatomy. So we use a lot of transgenic lines. This is a section through a 5-day-old zebrafish eye. This is the optic nerve. And we've got lines that allow us to use fluorescent reporters with cell type specific promoters, so we can see when these genes turn on and how the neurons are generated. So this is a nice readout developmentally, where we can see when things go wrong, for example, with migration or cell fate specification. Together with Beng Bui here at Melbourne University, Jiaheng has also set up the OCT for larval zebrafish that has been done overseas before and was published about 6 years ago. This is the setup for humans. So OCT is a non-invasive measurement of the ocular dimensions, including the retina. And so what happens is that light is shone into the eye and then reflected depending on the different layers. And this is what a 5-day-old larval zebrafish looks like. So you can see the lens and the retinal layers at the end of the eye. The other test that can be done, that is also done in mammalian models, also in Beng's lab, is electro-retinography. So we modified his mouse setup to work with larval fish. You can't actually see the larva because it's very small. But when we zoom in, Jiaheng managed to optimize this procedure to have more reliable results using a novel sponge tip electrode with a silver wire. And so he very gently touches the eye of the larva. We shine a light and we record basically the response in the eye. So we have a reference electrode under the wet sponge. We shine the light and we get a readout of the response of retinal neurons, which allows us to look at the visual performance. And finally, the Optomotor was established with Patrick Goodburn, who approached me with this whole idea, I guess about 5 years ago. And so for this, we have a zebrafish swimming arena with 6 swimming lanes. We put 50 larvae in each, and we show different stimuli. So here's an example where we have each of these dots is a larva. Each stimulus is shown for a minute. And you can see that we can change the spatial frequency, the temporal speed, so the temporal frequency, the contrast, and we run each stimulus 6 times in the trial. So the whole trial takes about 2 hours. And in between stimuli, we bring them back to the center. Zebrafish, we don't have to train them. They will automatically follow a visual stimulus if they can see it. And so you can see in this 30-second trial already that half of the fish from starting in the middle, they're now the center of their density is now in the same direction as the visual stimulus. So just by computing the center of their density before and after, we can compute an Optomotor index, and we can generate a contrast sensitivity function that shows which spatial frequency they see and how well they respond to that. And finally, Jiaheng realized very quickly that for some of the older fish, they run out of space pretty quickly in this setup. So he's now also developed a round Optomotor behavior test where an individual adult fish, we have to do this individually, can swim in the direction of the stimulus. So to first see if we could make this work and make sense, we just looked at neurodevelopment. We also wanted to see how sensitive these are. And we could see, for example, with the electro-retinogram, that 4, 5, 6, and 7 days post-fertilization, naive wild-type fish showed a maturation that we could pick up in the ERG. So this just shows light intensity. So the higher the flash, the higher the response. And we can see here when we overlay at the highest light intensity, the graphs, that at 7 days, the system has what we call matured, because the response is greater, but also faster. So the retinal neurons are working, the circuitry has been refined. We then looked at visual experience as a test to see if we can see any differences. And here's the data from the anatomy and the optomotor. So in the anatomy, we looked at postsynaptic density 95. This is an antibody stain. And we just quantified the density of synaptic puncta. And this is just merged with the DAPI layer. And we focus on the inner plexiform layer here, which is one of the two key synaptic layers. And we could see that, interestingly, not only did we see a refinement in the variability between 5, 6, and 7 days, but we could also see that animals that had been swum at day 5 and 6 as well performed, had an even higher synaptic density than those that saw the Optomotor test for the first time. Now, to distinguish whether that's a learning experience of being in the arena and swimming with the stimulus, we also had a cohort that just looked at static non-moving images on days 5 and 6 for the same 2 hours. And we found that both of these showed the same improvement. So we know anatomically that there's refinement if they've had visual experience rather than being just in a dark incubator. To see if we can translate that into some of the other phenotypic levels, we performed the behavioral test. And indeed, we could see these are the contrast sensitivity function, naive shown here in black. And more importantly, here the two ones at the top are the ones that either saw the static stimulus on days 5 and 6 or the moving stimulus in the arena. And both of those had a significantly higher amplitude. So we saw that correlation between the phenotypic levels, as well as we're able to demonstrate that the different pipeline components could pick up these subtle developmental refinements. What we're really interested in is to use this pipeline to look at novel gene candidates. So the first one we looked at came out of Pat's study. This is from his postdoc with John Mullen in England, where they used normal cohorts of humans and did a whole battery of different visual tests. His work was focused on contrast sensitivity. And here we can see in this Manhattan plot that he found significant SNPs associated with differences in contrast sensitivity. And this is just a position along the different chromosomes. We focused on this one here on chromosome 1, which is called PDZK1. And we do know that PDZK1, primarily from hippocampal culture studies, is associated with a lot of the synaptic receptors on the postsynaptic side. Interestingly, it is also found in a region of chromosome 1 on arm Q that has previously been identified as a hotspot for autism spectrum disorders. So we were interested in seeing what our pipeline could reveal about PDZK1. So in the Optomotor response, which mimics what he would have done with the human cohorts where he performs behavioral tests, we generated CRISPR mutants, and we could see that the PDZK1 knockouts actually performed better. And this is exactly what he saw in his human patients, that it's a SNP that changes an A to a G. And in one of the cohorts, you can get increased, or depending which one you use as your control, you get a change in contrast sensitivity. And we also know that enhanced spatial frequency tuning is one of the, not a hallmark, but one of the observations that does occur in schizophrenia and autism spectrum disorders. So at the ERG level, we found a reduction in the, this was unexpected, we found a reduction in inner retinal function, but it does actually also mimic what occurs in some cases of familial schizophrenia. And so when we look at amplitude, time, this is photoreceptor function, which is very small in zebrafish. B wave is this inner retinal function. We can see the main difference was this amplitude at different light intensities. We then thought we're going to now use the anatomy to figure out exactly why this happens, because that's going to be the most sensitive phenotypic assessment. And so we looked at all of the synaptic markers that had previously been identified, expecting a change in density or a loss of clustering. I tell you straight away, this is not what we found. We basically found no difference. And so it had been associated with serotonin receptors. So we looked at the synapses formed by serotonin, and again, wanting to know whether it's perhaps the association between the receptors and the postsynaptic density clustering proteins that is changed. We didn't see any changes. And Jaheng took this to older animals, thinking that maybe a phenotype would come up later, and we saw nothing. And basically the same is true for NMDA receptors and CRFR receptors, which had also previously been shown to be co-localized in hippocampal culture with PTZK1, and we saw nothing. So our conclusion was that there's no function in protein clustering, but we don't know how that then affects the functioning of these synapses. What was interesting from this, and completely opposite to what I expected, was actually that we now think that for different diseases, the different components of the pipeline might be informative. So it's not that just because anatomy has the best spatial scale, that's going to be the one where we see the most obvious effect. And so just briefly, I'll take you through some a current project where we're using this pipeline. We're actually focusing just on a couple of components. This was run by a master's student, Nicholas, in the lab, and it's a collaboration with Livia Carvalho, Carla, and Paula from the Lions Eye Institute. So it starts with a meta-analysis of human gene candidates that was published a few years ago that identified, again, over 100 novel components that showed genetic association with specifically childhood myopia, which is early onset, progresses more severe, and also has more severe secondary risk factors such as retinal detachment. So without going through the details, we can see a whole bunch of new genes. They're expressed in different cell types. They're also expressed in extracellular matrix. And we know that with myopia, the key ocular phenotype is that your eye is too big for your optics. So the light is actually focused in front of the retina, neuroretina that lines the back of the eye. So we wanted to see how visual function might affect the growth of the retina. Our approach is transient gene knockdown, so we can inject 500 to 800 embryos on the first day. And then we're doing the Optomotor response where we can have 50 larvae in each lane. We do 4 morphin lanes and 2 control lanes for every trial. And we do triplicates of the trials. So we've been able, in the 1 year that Nicholas actually had access to the lab before lockdown, we have been able to screen through 25 genes. Our initial aim was for 40, and we're hoping to get through some more. And just to show you very briefly some of the phenotypes, here are 4 examples of 4 different genes showing this Optomotor spatial frequency tuning responses. So when we knocked out GRIA4B, we could see that we had a reduced amplitude in the morphins, and this also occurred with RDH5. But otherwise, the fish were able to see the same kind of spatial frequencies. With FMP1 and GLIS3, we were able to see a shift of spatial frequencies. This actually also had a reduced amplitude and a poorer response at high spatial frequency. And this is what we initially expected for myopia. So this means that fine-scaled spatial frequencies, so skinny stripes, for example, cannot be distinguished by these fish. And this is what myopic people experience when they look at distance, where, for example, individual leaves on a tree start merging into just a blurry green blob. So we're using this, and we now have 7 candidate genes out of the 25 that we think are worth pursuing further by generating, for example, CRISPR mutants. Because a lot of these are developmentally important, we're also establishing a somatic CRISPR system where we're knocking out the gene using eye-specific Cas9 drivers. So they're only affected in the eye, but not in the rest of the central nervous system. And finally, Jaheng is now combining. Myopia actually has a massive environmental component, and we see this in high populated Asian cities where a lot of people live in small apartment blocks, a lot of reading, a lot of screen time, a lot of near work, and the prevalence of myopia has really risen within a generation. So for this, Jaheng is setting up dark mirroring has been shown to cause myopia. but he's also setting up different spatial frequency ranges that mimic what you might see if you're confined to an apartment or near vision, versus being able to see a whole range of different types of visual inputs. And the other one that's really interesting is using LEDs with restricted light wavelengths, because different wavelengths of light have different focal levels. There's a hypothesis that the exposure to longer wavelengths will induce a growth of the eye, which is sort of a myopic phenotype. So by only raising them, for example, in red light, we might generate a myopic eye. And similarly, we're knocking out the photoreceptor opsins for blue and green wavelengths, for example, to see if we can have the same, so if the same response is driven by that visual input. So far, he's only got data for the dark rearing. And for that, he was able to show that after 4 weeks, we get significant changes between the normal in black and the dark reared in blue. Now, this he did with an OCT setup, so measuring the ocular dimensions, which is how we would usually define myopia in a human clinic. And we initially started with axial length, so the size of the eye, but we found that there was no difference because the dark reared fish are actually overall smaller. So the body weight is smaller, the size is smaller, and that's probably reflective that they're actually very visual and they use their vision to catch food. So even though we adjusted the feeding to give them more time and turn the pump off while they're feeding, they just don't grow as well. So instead, we're now looking at the ratio between retinal radius and lens radius. So retinal radius as a measure of lens radius, which is a measure of ocular. And we're also starting at the curvature of the lens, which will affect how the visual system, how the visual input is focused. And the next plan is then to take some of our gene candidate CRISPR mutants and combine them with different environments to see synergies and links between the genetics and the visual input. And so with that, most of the work shown has been done by the master student Nicholas and Jaheng, who's, as I said, now continuing the environmental work as his PhD. And in collaboration, the pipeline was set up with Beng and Patrick, and the myopia work is done with my colleagues from the Lions Eye Institute. Thank you.
Thank you very much, Patricia. A lovely talk. So I think Julian's got a couple of questions. I might hand you over to him so he can ask those.
Oh, okay, thank you. Great talk, Pat. Yeah, like I just mentioned in the comments, great to see that you can herd zebrafish, but we still can't herd cats. But that's a remarkable Optomotor experiment. I would like to refer my questions to PDZK1, which was Patrick's SNP discovery. Was there any feature of the GWAS SNP that might be informative about how that would indicate the gene?
Oh, you just broke up.
Okay. Is PDZK1 relevant? The GWAS SNP, is it functional? So has it led to an inactivation of the gene or lowered expression? And how does that inform what you see in the mutant mice studies, fish studies? Can't shake mice.
That's a big what's the word I'm looking for? That's something that is not a limitation of the system, but that we need to keep in mind. The SNP is in the promoter region, and we actually don't know how it changes gene expression. So with our brute force approach of knocking out genes, that's not necessarily what the humans will have. In some cases, we actually, with the myopia study, had an improved visual response when we knocked out a gene. So we think that in the human, there might actually be an overexpression. So it's literally just, is this gene involved? And then the finer scale analysis needs to
absolutely. To be done. Yeah, I agree. It shows that the gene is relevant to visual system. Also, the point that perhaps PDZK1 might be relevant to muscle development in the event that they have inability to swim in response, but that's just a comment. Mike, can I hand it to you to handle questions for this session? That would be good. Thank you.
Yeah, there's one final question here, Patricia, from Ethan. Is there a way to be sure that your oculomotor phenotypes, especially the reduced phenotypes, are perceptual rather than sensory motor or motor?
It's the first thing the reviewers ask. Yes, so we've gotten together. I didn't have time to show this. We now use Ethan, you might know the Zebra box. It's automated system where we put well plates in with individual fish.
Right.
And you can activate them in different ways. You can just have them swimming in the dark under light. You can have a light stimulus to have a burst swimming. And so we've done the tests to look at, and it automatically calculates for you swimming distance, swimming burst, swimming duration, time, etc. Right. And so we've used that to make to show that they can still swim. So we see no differences. And also to see if it's a sensory thing. This is obviously, that's non-visual. Sure. We've also swum them with ethanol, which activates that sort of swimming. And again, they perform exactly like the controls. So we cannot rule out, because part of the visual system is that there is conservation across the central nervous system. So it could also have effects in the spinal cord neurons.
Sure, sure. Okay, yeah, really nice work, Patricia. Thank you. I look forward to talking more. We're doing a lot of the same stuff here, so this is great. I look forward to that.
Righto, fantastic, Patricia. Thank you again. I think we can move on to our third speaker for the session, which is Victor Borrell.